Hello student. Now, as in the last class we discussed about the introduction of biotechnology, right? So today we're going to discuss about the restriction enzyme. Restriction enzyme. Now, these are also said to be molecular scissor. Now, what does it mean? It means that they're going to cleave, they're going to cut. For example, this is your DNA duplex, okay? Double-stranded DNA. And you want to cut at a specific point, okay? Just say that this is your DNA molecule, a long DNA molecule, okay? And you want to cut, just say from here to here, from this point to this point, okay? So this restriction enzyme is going to cleave, cut at a specific point called, and we call that as palindromic sequences. Palindromic sequences. Okay, what does it mean? Okay, the meaning of palindromic sequences is, for example, nisin, N-I-S-I-N. Okay, a name. Okay, now just see N-I-S-I-N. Now you read from backward, N-I-S-I-N. Okay, or we can say as Malayalam, M-A-L-A-Y-A-L-A-N. See, M-A-L-A-Y-A-L-A-N. Okay, that read same from the forward and from the backward also that read same. Okay, so that is said to be palindromic sequences. Let me write one sequences. Now, don't think that those names are there in the DNA, okay? I'm just giving an example. What is the meaning of palindromic sequences? If I write G-A-A-T-T-C, then this is G, the opposite strand will be G-A-A-T-T-C, complementary strand, right? So if this is from 5 prime, it ends with 3 prime. So this is 5 prime. This is 3 prime. So if you read from forward, okay, just say this is forward and this is backward. It is same, right? G-A-A-T-T-C, G-A-A-T-T-C. Okay, because the complementary strand of cytosine is guanine, thymine is adenine, thymine, adenine, adenine, thymine, adenine, thymine, guanine, cytosine, right? So these are said to be palindromic sequences. Okay. Now, in those sequences, okay, these restriction enzyme are going to cut. Let's discuss its functions. Okay, why they are said to be molecular scissor? Because they cut at a specific point. Each restriction enzyme, different kinds of restriction enzyme have different palindromic sequences. Let me give an example. The first restriction enzyme, that is HIN2. Okay, it was Amber. Okay, these scientists have seen restriction enzyme for the first time in E. coli. So in E. coli, Amber had observed, okay, as a restriction enzyme in E. coli. Okay, it was Hamilton Smith. Okay, it was Hamilton Smith who had discovered this HIN2. Okay, HIN2 from HIN2 is the name of restriction enzyme. Okay, an example of restriction enzyme. So HIN2 was the first restriction enzyme which was isolated by Hamilton Smith. Okay, in Haemophilus, Haemophi, Haemophilus influenza. Okay, Haemophilus influenza. Okay, so this is a microorganism. Okay, so HIN2 was first isolated. It is known as the first restriction enzyme that has been isolated. Likewise, in your the textbook, okay, now it's more than 900 restriction enzyme has already been isolated, okay, from 230 strain of bacteria. Okay, 230 strain of bacteria, different kinds of bacteria, different types of bacteria, okay, different strain, okay, around more than 900, okay, restriction enzyme has been isolated. So this restriction enzyme is going to cut all the restriction enzyme has a specific point, has a specific palindromic sequences. Okay, let's take an example. Eco R1 is also one kind of restriction enzyme. Okay, so when I say Eco R1, it is going to cut at GAATTC. So opposite strain is, complementary strain is GAATTC. Now this will cut in this position, okay, and this will cut at this position between the bases. Okay, we're going to discuss on this. Now if we cut here, then, and if you cut here, so we're going to get this, right, like this, and the upper part, see, like this. Okay, likewise we have HIN2. It is going to cut at A G C T T. Okay, A G C T T. Now it is going to cut here. So this is A G C T T. See, it's a palindromic sequences. Okay, so it will cut here. So all the restriction enzyme, they have a specific point. In a specific palindromic sequences, they will cut. But between the bases, it means that they will cut both the strain. Okay, so that was asked last year in 2019 mid examinations. Okay, they asked about the features of the characteristics or the functions of the restriction enzyme. So they cleave both the strain. Both the strain means they will cut in this point where there is a guanine, for example GA. Okay, between the GA they will cut GA. For this HIN2 they will cut between the 89 bases A and A between them. So just say this is for 5 prime to 3 prime, 3 prime to 5 prime. It means that they are cleaving at both the strain, right? But if you take an example of endonuclease and exonuclease, they cleave at only on one strain, right? That is single strain. But you can see that restriction enzyme, they cleave both the strand. Okay, that is at a specific point. Okay, so same as this, they will cut here, and you get this as a one strand, one end. So this is one end, this is another end. And this type of ends are called sticky ends. Okay. Likewise, we have so many restriction enzymes. I just took 2 examples. Okay. Now, there's some restriction enzyme. Just say that this is G, G, C, C. So this is G, G, C, C. One GGCC, GGCC. Okay. So just say that this is one strand. So if I cut in the middle, then what will I get? GGCC, one strand. Okay. And one end, another end will be CCGG. Such type of end reductions are said to be blunt end. Blunt ends. Okay, so some restriction enzymes will produce sticky ends, and some restriction enzyme will produce blunt end. Okay, so this is about the restriction enzyme and its important functions. Now, we're going to discuss about the nomenclature, how to name the restriction enzyme. Okay. Now, naming restriction enzyme is very important. That is nomenclature. I'll say nomenclature. Let's take one example. Okay. If I write equal R1, so this is the first letter, the capital letter. This is E represent. Okay. Like just say that hint2. Hint2. Just say. So this first letter represent the genus. Okay, from which bacteria? Okay, which genus it belongs? Okay, and the second two letter likewise. See, this is genus. The second two letters, remaining two letters, small letters. Okay, so these are said to be the species. Okay, so second letter represent your species. Okay, and the first letter represent your genus. So this is species. Represent species. Okay. Now the next letter R. Okay, R represent the name of the strength. Strength. Name of strength. So likewise, this D represent the name of the strength. Okay. Now, this Roman number 1 or Roman number 2 is said to be the order of the strength. Okay, the order. Now, this is what you have to know. Okay, this was also asked in your NEET examinations. Okay, so this is very important. The discovery of this restriction enzyme, which was done by Amber, who have seen Amber have seen this restriction enzyme in E. coli, okay, as a defense mechanism. Okay, so when a virus, okay, when a virus attack to bacteria, when a virus attack bacteria, the virus will release their genetic material, right? So just say that this is bacteria, and there is a the viruses have introduced this genetic material. Just say that this is virus. This virus have introduced this genetic material in a bacterial cell. So there is a molecular scissor, restriction enzyme. What they do is that they cleave this, they cut this viral DNA. So if this viral DNA was cleaved by the restriction enzyme, then the virus will not multiply. They will degrade, okay? They will die. They will not able to multiply itself because its genetic material has been destroyed by the restriction enzyme. So there was one question that why bacteria having a restriction enzyme cannot cleave its own DNA. So that has to be understood, okay? Why? Why they cannot cleave its own DNA? They also have their own genetic material, okay? Because they go for certain modifications, and that is called methylase. What they do is that within their system, they started adding methyl group in their own DNA, okay? And that is the reason that they recognize. They know that it is their genetic material. But when any foreign material enter inside the bacterial cell, they cleave, they destroy. Okay? Then it was Hamilton Smith who had isolated the hint2 from Haemophilus influenza. Okay? So that's why they are called as the molecular scissor. The important tool that is required in a biotechnology is restriction enzymes. Okay? Now, in next class, we're going to discuss about the processes of rDNA technology. I'll just write what are the topics that we're going to discuss. Okay? First is isolation. The step that is required in biotechnology is isolation of DNA. We're going to discuss how DNA will be isolated. Second is that we are going to cleave the DNA. The DNA which was isolated has to be cut into by restriction enzymes. Okay. Next is said to be gel electrophoresis. Next step. The fourth is called the transfer of DNA, target DNA to host to vector, or we say transfer of DNA to introduction of DNA to vector, and then we have introduction into host, into host cells, okay, and then we have processing, okay. But these 5 important steps: isolation of DNA, then cleaving of DNA by restriction enzymes, then we have to separate it, the DNA fragments, with the help of gel electrophoresis, then we have to transfer the DNA to vector, then we have introduction in the host cells, then we're going to discuss about PCR, and then we're going to discuss about the bioreactors. So this is the topic that we're going to discuss in the next class, okay? Before I end up, one thing you have to know that your rDNA, I'm telling you again and again, your rDNA is the vector, your vector and the DNA fragments, okay? Vector and the DNA fragments. Why do we need a vector, okay? Because we need some vehicle to transport our gene of interest, okay? And this is also said your chimeric DNA. This term, chimeric DNA, okay? Now, in the next class, we're going to discuss all those things. I hope that all the students, you observe the first part and the second part video properly, so that in order to understand this concept in the next classes. Thank you, class.